Hello students, this is video 3 of gametogenesis. In the last video, we talked about the process of spermatogenesis in detail. Today, I'll be discussing with you the structure of the mature spermatozoon. I have divided my presentation into 4 sections. First deals with the structure of the mature spermatozoon, and I'll be covering the topics of head, neck, middle piece and tail. Then I'll be talking about sperm pathology, then types of sperms, and last I'll be covering the functions of the sperm. So let's talk about the structure of a mature spermatozoon. Now spermatozoa are highly specialized. The spermatozoa of aquatic invertebrates which shed them to the water are primitive, whereas those of animals with internal fertilization are quite elaborate. Now let's look at this diagram of the mature spermatozoon. As you can see, outside it is covered by a thin plasma membrane. At the tip is present the acrosome. Below it is present the nucleus, and it is surrounded by fibrous sheaths. This whole structure is called as the head. Then there is the neck or the connecting piece, which has the proximal centriole, which leads into the distal centriole. The next section is the middle piece, which in its center has the exoneme or the axial filament. There is the distal centriole, and then there is the mitochondrial spiral around it, covered by the cytoplasm, the cytoplasmic sheath, and then these fibers that are covering it. Below it is present the tail, which is covered on the outside by thick fibers, and the axoneme or the axial filament continues inside this tail. Now let's talk about each of the substructures of the spermatozoon in detail. First is the head region. This is the diagram showing sagittal section through the head of the mammalian sperm. As you can see, again on the outside there is present a plasma membrane. Then there is the acrosome present here, which consists of 2 segments: the principal segment from here to here and the equatorial segment, which is present here. The acrosome contains the acrosomal contents. It will have an outer membrane and an inner membrane. Below it is present the nucleus, enclosed by the nuclear envelope. This region is called as the post-acrosomal region. So the head of the sperm may be of different shapes. It may be rounded, it may be oval, maybe rod-like, maybe lance-shaped, spiral or hooked in different kinds of animals. The plasma membrane which surrounds the head has an acidic protein which is called as anti-fertilizin. This is a species-specific protein receptor of fertilizin protein, which is present in the jelly coat of unfertilized eggs. So anti-fertilizin is present in the sperms, while fertilizin is present in the eggs, and both of these fuse with each other during fertilization. They help in they help the sperms to bind only with the eggs of its own species. The nucleus in the head is very compact, as we talked about earlier. It will have a condensed DNA and basic proteins. It does not have RNA, acidic proteins, or any kind of nucleolus. The acrosome forms a cap over the nucleus, and the space between the acrosome and the nucleus is called as the subacrosomal space. When fertilization occurs, this subacrosomal space develops into a rigid acrosomal filament which contains microfilaments. Because of its origin from the Golgi complex, the acrosome can be regarded as a specialized lysosome. And as you know, lysosomes have specific enzymes. So this also contains hydrolytic enzymes which are called as sperm lysins. The enzymes like acid phosphatase, cathepsin, hyaluronidase, etc. In mammals, the acrosome also contains acrosomin, which is the combination of enzyme hyaluronidase with acrosin, which is a zona lysin. So this penetrates the zona radiata and pellucida. So it is used to penetrate the egg membranes. The apical segment of the acrosome is quite small and inconspicuous in man. The equatorial segment of the acrosome contains the outer and the inner acrosomal membranes, which are linked by bridges. It is the first site of contact between the sperm on the egg. This persists during fertilization. The rest of the acrosome is lost during the acrosome reaction. So only the equatorial segment of the acrosome persists during fertilization. So going back to the diagram, this is the area where the sperm first makes contact with the egg. This is the area which persists even after fertilization. This all rest is lost. The post-acrosomal region of the head contains a dense layer beneath the plasma membrane. So this is the post-acrosomal region which contains the dense layer. Now we'll talk about the neck. It is quite short and it contains a convex connecting piece lying in a concavity at the base of the sperm head. So this is the connecting piece that is present here, present in the concavity. It articulates to the head by very fine filaments, and it consists of 9 segmented columns which join the outer dense fibers of the tail. In mammals, the neck contains 2 centrioles. The proximal centriole takes part in the first cleavage of the zygote by forming the mitotic spindle, while the distal centriole gives rise to the axial filament of the sperm. So in mammals, both the centrioles have good functions. The proximal one will form the spindle when the fertilization has taken place. So the zygote is formed and then the zygote starts to divide further, whereas the distal one gives rise to the tail of the sperm, the axoneme or the axial filament of the sperm. The centriole itself contains 9 sets of microtubule triplets arranged in a ring. So these are the microtubule triplets, 9 in number, arranged in a ring form. Now we'll talk about the middle piece of the sperm. The middle piece of the sperm on the outside has a mitochondrial sheath formed by the mitochondria. Inside it are present the outer dense fibers which then connect with the neck, and inside it is present the axoneme or the axial filament. So there are 3 structures. Outer is the mitochondrial sheath, then the outer dense fibers, then the axoneme or the axial filament. The middle piece varies in shape in different species. It is cylindrical in case of human sperm. It contains mitochondria in a very thin layer of cytoplasm. The mitochondria are tightly coiled end to end in a helical chain, as I described in the diagram, forming a sheath around the axial filament. Now, we know that mitochondria has oxidative enzymes. These enzymes will help to oxidize endogenous or exogenous substrates. So endogenous substrates means which are present inside the sperm itself, like the phospholipids, and exogenous substrates are those which are present in the fluid that surrounds the sperms, that is having fructose. Both these substrates are oxidized by these enzymes so as to provide energy for the movement of the sperm. Now the axoneme also starts within the middle piece, so its proximal part is present inside here also. It consists of two central microtubules having the enzyme acetylcholinesterase surrounded by a ring of 9 doublet microtubules having ATPase enzyme. They are surrounded by outer dense fibers which contain enzymes spermatozoon, succinic dehydrogenase, ATPase, etc. Just let me go back to the diagram. Here you can see this is the axoneme or the axial filament. This will have two central fibers and 9 doublets of microtubules. The central fibers will have acetylcholinesterase enzyme and the outer fibers will have dynein arms having ATPase enzyme. The outer dense fibers also contain enzymes as I described. The spermatozoon, the succinic dehydrogenase and the ATPase. Now where the middle piece ends, that structure is called as the annulus. After the annulus, the tail region starts where the axoneme is then covered by a fibrous sheath. Now this is the cross section of the axoneme. As I told you, the axoneme consists of 9 doublets of microtubules having dynein arms and 2 central singlets. They are themselves covered by the plasma membrane. Each doublet consists of 2 tubules, the A tubule and the B tubule, and each tubule further contains 13 microtubules. There is radial spokes that join the doublets with the central sheath. The 2 singlets are themselves connected by a bridge. The doublets are also connected with each other by interdoublet links having nexin. Each doublet, as I explained, has 2 subtubules A and B, and each subtubule A will have dynein arms, the outer arm and the inner arm. These are further linked by a membrane link to the plasma membrane. So this is the transverse section of the axoneme or the axial filament, which is present both in the middle piece and the tail. So the tail is very long, slender and tapering. A fine thread, the axial filament or the axoneme, arises from the posterior centriole and transverses the middle piece and the tail. The axial filament is similar to the flagellum. As I explained, it has 2 central microtubules surrounded by a ring of 9 doublet microtubules. The central singlet microtubules have gaps between them but are connected by a double bridge and are surrounded by a common central sheath. Each singlet microtubule consists of 13 protofilaments arranged in a ring. The peripheral doublets consist of two subtubules A and B. Subtubule A is complete, having 13 protofilaments, while subtubule B is C-shaped and consists of 10 protofilaments. Going back to the diagram, here you can see this is having 13 protofilaments, this is having 13 protofilaments. The subtubule A is complete, having 13 protofilaments, but the subtubule B is C-shaped and has only 10 protofilaments. The subtubule A gives off inner and outer arms, and the arms run clockwise. A radial spoke extends from the central sheath to each subtubule A, and it shows a thickening which is called as the spoke head. So in the diagram, if you see, this is the spoke head, and this is the radial spoke connecting the doublets with the central sheath. All doublets are themselves joined by interdoublet links, which are made up of nexin protein, and all the microtubules are formed of tubulin protein. The arms contain dynein 1 protein, which has an ATPase enzyme which transfers energy released directly to the doublets for their sliding movements, which help in the movement of the tail. So we look at the tail. As I told you, instead of the mitochondria, as in the middle piece, the axoneme is surrounded by the outer dense fibers, and then they are surrounded by the fibrous sheath. The fibrous sheath will have circumferential ribs present. So these are the C-shaped rings or ribs present. So the outer ring has accessory dense fibers, each running parallel to the doublet. These fibers are thicker proximally and then become thinner at the tip of the tail. The outer dense fibers are the motile elements, and they generate additional force for vibrating the tail, and they also provide the elasticity to the tail. So these are the outer dense fibers. They help in the movement of the tail. They also help to give elasticity to the tail. The fibrous sheath also covers the tail in the principal piece. It consists of two longitudinal columns connected by ribs which are hemispherical. The longitudinal columns are anteriorly joined to the outer dense fibers. As we go towards the tip of the tail, this fibrous sheath thins posteriorly and ends a short distance from the tip. This part of the tail is called as the end piece. So here you can see the diagram of the sperm. This is the head of the sperm, the connecting piece or the neck, the middle piece, the tail. The tail will have the principal piece and the end piece. If we look at the section here of the mid piece, as you can see, mitochondria are present, plasma membrane, cytoplasm, then mitochondria, then outer dense fibers, then the axoneme, the 9 doublets and the 2 singlets. If we look at this principal piece, again cytoplasm present, plasma membrane is present. Here you can see one C-shaped one longitudinal column here and one longitudinal column here, and they are joined by these ribs. These are again the outer dense fibers as were present here, and inside again the 9 doublets and the 2 singlets. If we look at the end piece, here no fibrous sheath is present, no dense fibers are present, only the axoneme is present, the 9 doublets and the singlets. So it is quite thin as compared to the principal piece of the tail. Now let's talk about sperm pathology. If dynein arms are lacking in the outer doublets, this results in immotile sperms. Though the sperms look morphologically normal, but these dynenarms, if they are absent, the sperms will be immotile. People with this defect, having which is called as the immotile cilia syndrome, are infertile, and this occurs due to an autosomal recessive mutation. So another thing is the 9+2 pattern, the 9 outer microtubule doublets and the 2 inner central singlets. This pattern is essential for the motility of the sperm. If there is any deviation here, this will again have atypical motility, not proper motility. So those will also be sterile. If there is some kind of problem in the outer dense fibers also, as I told you, outer dense fibers are related to motility of the sperm, again the individual will be sterile. The sperms will be unable to move or to fertilize the eggs. Here is the scanning electron microscope of various kinds of pathologies. In this A figure, you can see there is a dysplasia of the fibrous sheath. So there are two tails, very short tails, which are present. This is the normal structure. In C and D, you can see there is difference in the structure. In C, there is two, this is called as ciliary dyskinesia. The fused complete and incomplete axonemes are there because of the neighboring spermatids did not separate, and there is also lack of some kind of dynein arms here, and there is distortions in the fibrous layer also. In D, there is no central singlets, so this is again is a pathology, a problem. Now these sperms EFG, there is some kind of problem, abnormality with the fibrous sheath that is surrounding the axoneme. So again, this will result in sterile sperms. Now what are the various types of spermatozoa? There are two main types, the flagellated and the non-flagellated. In case of toadfish or Opsonus, there are biflagellate sperms. In case of ascaris, crabs, lobsters, centipedes, millipedes and mites, there are non-flagellated sperms. These are the diagrams of different kinds of sperms. A and B, these are the sperms of teleost fishes. C and D belong to birds. E and F is belonging to snails. The G sperm, this one is amoeboid. This is present in ascaris. H belongs to an annulate. I, bat. J in an opossum. This K type is in rat. This L type sperm is present in salamander. M and OP all are crustaceans, all are non-flagellated. And next we move on to what are the functions of the sperm. There are three main functions. First, it activates the egg to start development. As soon as the sperm fuses with the egg, it activates it. This egg has entered into suspended meiosis. So the egg is present at the stage where meiosis has not been complete. As soon as the sperm fuses with the egg, it stimulates the egg to complete its meiosis. It contributes the male's haploid set of chromosomes to form the zygote. And third, it provides the centrioles to the zygote. As I told you earlier, the proximal centriole of the sperm will help in the division of the zygote, the first division or the cleavage of the zygote. So these are the three major functions of the sperm. These are the references that I consulted to make this video presentation. In the next video, we'll talk about the role of the accessory somatic cells during spermatogenesis. Thank you.